So this video lecture is going to be on cell communication. And right off the bat, you see two human-like figures and those old-fashioned cans with the string, and you can talk and listen through them. And I'm using that as the communication. So here we have cell signaling, and for an example, I'm using animal cells. And they communicate by direct contact, secreting local regulations, and long distance. Now, direct contact, I mean the gap junctions. And for secreting local regulations, or regulators, as the growth factors and neurotransmitters. For long distance, I'm talking about their hormones. Here you see two faces and they're talking. At the top left corner you see FBI agents, and on the bottom right corner you see walkie-talkies. And so the FBI agents use the walkie-talkies to communicate with each other, as seen right here, how they communicate. They talk through the walkie-talkies, and bam, they're all together. Now, there are 3 stages of cell communication, and 1, it's reception, 2, transduction, and 3, response. Now, everyone's seen this picture before in our bio packets. But I'm just reusing it because it's very helpful. Reception is the detection of a signal molecule, the ligand, coming from outside the cells. Transduction converts signals to a form that can bring about a cellular response. And response is a cellular response to the signal molecule. So in this picture, on this slide, we clearly see where the cell, the cytoplasm, plasma membrane, but we see the steps: 1, reception; 2, transduction; and 3, response. With the reception, there's 2 types. You have the plasma membrane receptor, which is a water-soluble ligand, and you have an intracellular receptor, which is the cytoplasm, the nucleus, and that's the hydrophobic or small ligands. Ligand binds to the receptor protein, and then the protein changes its shape, and then it initiates a transduction signal. Second, we have transduction, and in that, it's a cascade of molecular interactions, relay signals from receptors to target molecules. There's also protein kinase, and that's an enzyme that phosphorylates and activates proteins at the next level. And then you have the phosphorylation cascade, and it enhances and amplifies the signals. We then go to the 3rd stage, which is response, and that regulates the protein synthesis by turning on or off the genes in the nucleus, which is the gene expression. And it also regulates activity of proteins in the cytoplasm. We have second messengers, and that's a small non-protein molecules or ions that can relay signal inside the cell. Oh no, apoptosis. That is otherwise known as the cell suicide. Apoptosis is a cell dismantled and digested. It is triggered by signals that activate cascade of suicide proteins. And this happens because 1, it protects the neighboring cells from damage, and 2, animal development and maintenance. This may be involved in some diseases, and interference may contribute to cancer. To show an example of apoptosis, I found um, this little picture, like of two, I guess you could say, cells, and so I'll just read them. The first one holding the gun right here to its head, it says, "I am done with my role here. It's time for me to die now." And the bottom guy is saying, "I will miss you, my altruistic brother." The bottom, it says, "Cells have predefined suicidal pathways that are activated in case of cellular damage, like I said before, or after accomplishment of a particular role." So that kind of sums up apoptosis.